Hi everyone, my name is Hannah Lee, and I will be presenting on bacteriocins against contact lens biofilms. Bacterial keratitis is a bacterial eye infection of the cornea, which is the outermost layer of your eye, and it's commonly caused by Pseudomonas aeruginosa and Staphylococcus aureus. And these two bacterial species are a part of the escape pathogen list, which are six bacterial strains that are extremely harmful and resistant to many antibiotics. Approximately 17% of the adults in the United States wear contact lenses, and amongst these contact wearers, bacterial keratitis is the most common eye infection. And keratitis can cause damage to the cornea, but can also eventually lead to blindness. And biofilms can form on the cornea as well as on contact lens surfaces, which include silicone and other plastic materials. Biofilms can be defined as bacterial communities composed of planktonic and sessile cells. Planktonic is defined as free floating, so it's labeled free swimming in the image below. And sessile is defined as stationary, so these cells don't really move after it's attached onto a surface. And antibiotic resistance is a very big problem globally, and greater than 2.8 million antibiotic resistant infections are reported every year just in the United States. And misuse of antibiotics, which include overuse and overprescription, has caused more resistance over the years, which has caused antibiotic resistance to continually be on the rise. One novel alternative to antibiotics is bacteriocins, which are defined as antimicrobial peptides that are produced by most species of bacteria. And compared to antibiotics, these bacteriocins have a narrow spectrum activity, so it kills specific strains rather than wiping out an entire microbiome. And one bacteriocin that we use in the Riley lab is nisin, which is produced by Lactococcus lactis. And nisin has shown a lot of efficacy against gram-positive bacteria, but also gram-negative bacteria. Um, by the FDA, it's been considered safe and non-toxic because it's been used as a food preservative, so it's not foreign to the body. And in the team that I'm in, Team Nissen against keratitis, we compare Nissen to polymyxin B, which is a commonly used antibiotic for bacterial keratitis. However, there have been high rates of resistance to polymyxin B, so there is a very high need for an alternative. And the purpose of my research is to observe biofilm formation on silicone contact lenses and test the efficacy of nisin against these biofilms that cause keratitis. And my hypothesis is that if there's an appropriate environment for bacterial attachment, then Pseudomonas aeruginosa biofilms will be able to form on silicone contact lenses and therefore be observed and quantified. And if Pseudomonas aeruginosa biofilms are formed, then nisin will have an inhibitory effect. So the methodology, I used a well plate that I incubated a cell dilution as well as contact lenses. And after 8 hours of incubation at 37 degrees Celsius, they were then treated with nisin at three different time intervals, 2 hours, 4 hours, and 16 hours. And then after those incubation periods, they were then transferred into another Eppendorf to be vortex and remove the biofilm. And then that solution was then plated to see if there were any colonies that would form. So on the left most picture is the actual well plate that I use. You can see that there are contact lenses as well as the cell dilution I used. And on the right is a picture of the contact lens post incubation. So you can kind of see the turbidity on the contact lens compared to a sterile contact lens, which would be completely clear. So the results on the left is the growth control, which was the cell solution before the incubation, the original incubation in the well plate. And on the right is the growth control after the 16-hour incubation. As you can see, the cells grew, and in an ample environment, in an ideal environment, there would be a lot of cell growth. And here are the agar plates that contain the solution of the nisin treatments. So you can see on the right, there is the 16-hour Nissen treatment, and in the middle is the 4-hour Nissen treatment, and on the left is the 2-hour Nissen treatment. And as you can see, there are no colonies on any of the agar plates, which shows that Nissen can kill not only in 16 hours, not only in 4 hours, but Nissen can kill in 2 hours. And the conclusion is that Pseudomonas aeruginosa biofilms can form on silicone contact lens surfaces, as well as Nissen at our neat concentration, which is 5.12 milligrams per milliliter, has a killing effect of 2 hours. So for future experiments, I would like to expand the strain numbers. I would like to test more Pseudomonas aeruginosa strains, as well as test the other common strain in bacterial keratitis, which is Staphylococcus aureus. And I would like to decrease the Nissen treatment time to under 2 hours, because we see that there are no cells that are present after 2 hours. So I'd like to go into 30 minutes, an hour, maybe even lower. And then I would also like to decrease the Nissen concentration. Right now, we tested it at 5.12 milligrams per milliliter, but I'd like to see if they can have the same killing effect with a lower dilution. And I would like to acknowledge the Riley Lab, especially our PI, Dr. Margaret Riley, as well as my team leader, Natalie Getzi. Thank you for watching.